This is preparation for lecture 8, and here we're going to be looking at earlier stages of development. We're going to actually look at the process of oogenesis, so how the egg is formed and how certain determinants are localized to particular locations within that oocyte. And this is a very beautiful picture. This is an egg chamber. So it's this long structure like this. And each of these sort of greenish regions that are stained for one protein are surrounded by red cells that are stained for a different protein. And this is an egg chamber. So the germ cells are in the center part here, and the somatic cells that surround them are called follicle cells. And there's a whole lot of follicles in this picture, and each one of them has one oocyte and some nurse cells that we'll talk about. And you can see from, this is like a timeline. So you can see the very earliest stages, and then later and later and later stages. This is all happening, multiple eggs are being produced over time. And you can just look at one of these ovarioles and see how the process develops. So we're going to look at that first. We're also going to, once we've talked about the formation of the egg chamber, we're going to be talking about patterning it, how it's patterned so that the anterior and posterior parts of that one giant cell are different from each other, and how the dorsal and ventral are specified as well. Okay, so the last couple of lectures we've been talking about the basics of development, and we've focused on maternal components, so the things, the proteins and the RNAs and so on that are put into the egg by the mother, and in that case, they're put in during the process of oogenesis. So this is a real photograph of a pair of ovaries from a female fly dissected out. And they look like a bunch of bananas inside a sort of a net bag. And if you look at it, you can't really get a good sense of the scale from this picture, but I used to work on oogenesis in flies, and many, many decades ago now. But when you remove them from the body, they're approximately the same size as the head of the fly, which gives you some sense of the relative importance. The main function of the adult stage of Drosophila is the reproduction. Their whole aim is to reproduce as much as possible, and so these enormous ovaries are how that's done. When you look in more detail, you can dissect away the little fibrous kind of tissue that's holding it together, and each of the bananas, if you like, in the bunch of bananas is an ovariole, and each ovariole contains a bunch of different egg chambers. So that's an egg chamber, that's one, that's one, that's one, and so on, and that's one. And each of them is, at the bottom is the most mature ones, and at the top, the little ones are the youngest ones. Yeah, and so let's see, I think that's all you need to know from this image. But we're going to be looking at this in more detail, because each of these, as I said before, is like a timeline. You can see sort of the process of development in a single ovariole, which is a very interesting thing. So the very first part is called the germarium. It's very tiny, and this is where the stem cells are dividing. For each division, one stem cell divides to produce one more stem cell, but one cell called a cystoblast that's going to give rise to the inside parts of all these follicles or egg chambers. So germarium, and then vitellarium. Vitellin is about the egg and is about yolk. And so this is where the egg is actually forming the oocyte, if you like. the yolk is being put in. And so in the very earliest stages, you have 16 cells, and they're surrounded by somatic cells. So these are not reproductive cells, they're supporting cells. And they take part in the signaling that does the patterning, but they're not going to form the germline. But they pinch off individual follicles or egg chambers, and in each of these you have inside 16 cells. One is the oocyte and 15 are nurse cells. And then a set of follicle cells on the outside. There's some rearrangements and changes that we'll talk about. And then about midway through the vidularium stage, you start to see the oocyte is growing a little bit faster than the nurse cell, so it's relatively just a little bit larger. And over time, this gets exaggerated more and more until you get to this is about stage 10, which I always remember because it looks like a shoe print to me with the toe here and the heel there. And at this point, the oocyte is almost half of the total inside batch of cells. And then over time, from stage 10 to the mature, stage 13, the nurse cells contract and they squeeze all their contents into the oocyte that then sort of grows and takes over the entire inside part of the egg chamber. And these nurse cells get degraded eventually. Okay, so let's look in a little bit more detail. So here in the very tip of that egg chamber, you have stem cells. Stem cells are self-renewing and they are able to form any structure. So in this case, the stem cell divides, it forms another stem cell, which then can go through this process again. We call this a self-renewing process. But one of the cells is different. It contains something different. And that sets it on its own path, which is to become a cystoblast. And the cystoblast goes through mitosis. The cells divide, but they don't completely divide. There's a little chamber called a ring canal that's a little a channel between those two cells. So they almost completely divide, but there's a little tube left that connects them to each other. Then there's additional 3 more divisions, and in all cases these are not complete divisions. The little ring canals are left behind, and so the most recently formed cells only have one connection to their neighbors, and the ones formed after that, like this one, will have 2. There are some most of them have either 1 or 2, but there are 2 cells that have 4 connections. So these are the 2 original cells shown here. This one is this one, and this one is this one. And there's a structure inside one of these 2 cells. It's called a spectrosome. It's microtubule based, and it's, if you remember some of your cell biology, it takes part in the formation of a microtubule organizing center. And whichever of these 2 cells has the microtubule organizing center in it is going to form the oocyte. And it can be recognized by the fact that it has this visible microtubulin-based structure in it, but it also is one of the 2 cells that has 4 ring canal connections to the others. So it has the maximum number possible of connections to the other cells. And you can see that through this ring canal, both of these cells can be accessed. Through this one, all of these can be accessed, and so on. If you imagine microtubules starting at this center, they can extend out and through and reach all of the other 15 cells. And these are nurse cells, and a lot of the materials that they contain are actually transported, directed into the oocyte, and in most cases, not all, but in most cases they are directed along these microtubules. So they're attached to motor proteins that take them towards the source of the microtubules. Okay. Um, and so that's the formation of the cyst, and then the follicle cells surround it. And then by about stage 4, you can see the oocyte is around the same size as the other cells. But by stage 8, the oocyte is a bit larger. It's about the size of 2 nurse cells. And follicle cells are beginning to rearrange. So there's some signaling and stuff that's going on in this region here that's shown by these different colors. We will be talking about this in a minute. And the cells are getting taller and thinner, columnar. They form columns when they're around the developing oocyte, but they get thinner and sort of wider, and they spread out more across the nurse cells. So there's lots of follicle cells around the oocyte, and over time there's relatively fewer follicle cells around the nurse cells. By stage 9, the oocyte is around a third the size of the total egg chamber. And by the time we get to stage 10A, which again, I remember stage 10 because it looks like a shoe print to me, then almost all the follicle cells are columnar. They're around the oocyte part. The oocyte is about half the size of the whole egg chamber. There has been some migration, so some of the cells in the front here have migrated through the nurse cells and have come to be positioned right here to specify the anterior part of the egg chamber. There's also some signaling we'll talk about that specifies posterior part of the egg chamber. And at the beginning of this process, the oocyte is kind of glued to the posterior pole by cadherents, cell cytoskeletal proteins. But then, and that helps with anterior-posterior specification, but then there's microtubule and other rearrangements that lead to the nucleus migrating up into what we would call the dorsal anterior corner, and then additional dorsal ventral specification happens here. So there's a number of different things that are going on that we're going to talk about. So this is what it looks like, and we will just cover a little subset of the actual signaling and so on that's going on during this process. At the later stages, the main thing that happens is that a large kind of dump of all the contents of the nurse cells takes place, making its way into the oocyte. So there's a lot of proteins. There's a little bit of probably yolk, just a little. There are RNAs and other molecules there. Some of them have been transported already into the oocyte and have helped with specification. But in the meantime, all the organelles, the ribosomes and so on, all that is needed in the oocyte because it's going to be very busy dividing once fertilization has taken place. And so as the oocyte grows, it kind of squeezes the nurse cells, and there are some fibrous proteins around here that also kind of contract. I don't know why I'm making a gesture with my hand. They kind of contract to squeeze the nurse cells. And the idea is that there's this huge flow of materials into the oocyte. By the end of the process, you have a mature egg. You have developed an eggshell, which is the gray on the outside. The follicle cells have basically disintegrated, but they have left behind an imprint on that eggshell, and they have developed, the micropyle at the anterior end, and they have anterior versus posterior, dorsal versus ventral. Here are your dorsal appendages and so on. So the follicle cells, they pattern the eggshell, they allow for the development of all these structures, and they leave an imprint on the surface of the egg that you can actually see, that I pointed out in lecture 6, I believe. Okay, so basically, as the egg chambers are produced